It's pretty mind-blowing when you stop and think about it. Every single living thing, from a tiny ant to a giant blue whale, starts its life as just one single microscopic cell. That's where every life story, including yours, begins.
But that begs a huge question, right? How in the world does that one cell become the trillions of cells that make up a complex creature like us? How does it all work? Well, let's dive right into that incredible story of growth and division.
At its core, the answer is actually really simple. A cell's main purpose is to become two cells. That's it. It's this constant, elegant cycle of growing and then dividing. This cycle is the engine that takes that single starting cell and builds an entire organism from scratch.
Now, scientists have a name for this, of course, the cell cycle. And you got to understand this is not a random process. It's more like a perfectly choreographed dance. The cell has to duplicate everything inside it, especially its precious genetic blueprint, the genome, before it can split into two new daughter cells. And every single step is under incredibly tight genetic control.
So, the first and, believe it or not, the longest chapter in a cell's life is a stage called interphase. For a long time, people called it the resting phase, but wow, that couldn't be more wrong. This is when the cell is working its hardest, getting everything ready for the big moment of division.
And what's just wild is how much time is spent just on prep. I mean, take a look at this diagram. See that massive slice of the pie labeled interphase? That's more than 95% of the cell's entire life. The actual division part, the M phase, is just that tiny little sliver at the end. It's all about getting ready for the show.
And this prep phase has its own 3 acts. First up is the G1 phase. Here, the cell just grows, it gets bigger, and does its normal cell stuff. Then comes the really crucial part, the S phase, where S stands for synthesis. This is when the cell makes a perfect copy of all its DNA. It literally doubles its entire genetic library to make sure both new cells get a complete set of instructions. And finally, there's the G2 phase, which is like the last-minute checklist, making a few more proteins, growing a little bit more, and getting ready for the main event.
So to give you the sense of the timing, for a typical human cell growing in a lab, this whole story, from start to finish, takes about 24 hours.
But get this, in biology, things can move at wildly different speeds. A simple use cell? It can blast through the entire cycle in just 90 minutes. It's just an amazing example of how diverse life is, even at this tiny scale.
And here's a really interesting twist, one that has huge implications for us. Not all cells are constantly dividing. Some of our most critical cells, like our brain's neurons or the muscle cells in our heart, actually leave the cycle. They enter this sort of non-dividing retirement state called G0. They're still alive and doing their jobs, but they've stopped reproducing. And that's exactly why an injury to your brain or your heart can be so permanent. Those cells just don't replace themselves.
All right, let's get to the climax of the story, the M phase, or what we call mitosis. This is easily the most dramatic part of the whole cycle. It's like a brilliant biological engine designed for one single purpose, creating perfect clones. This is what fuels our growth and what repairs our bodies every single day.
The key thing to remember about mitosis is that it's an equational division. That's just a fancy way of saying the number of chromosomes stays equal. So if the parent cell has 46 chromosomes, the 2 new daughter cells will each have exactly 46 chromosomes. It's a perfect copy and paste operation.
This whole thing unfolds in 4 beautifully choreographed acts. First, in prophase, all that messy, stringy DNA coils up super tightly into those classic X-shaped chromosomes we see in textbooks. Then, in metaphase, all of those chromosomes get pushed and pulled right to the middle of the cell, lining up in a perfect row. Next up, anaphase. The 2 halves of each X-shaped chromosome are ripped apart and dragged to opposite ends of the cell. And finally, in telophase, a brand new nucleus forms around each of those 2 groups of chromosomes. Just like that, you have 2 command centers for 2 new cells.
But hey, dividing the DNA isn't the whole story. The cell still has to split its entire body, the cytoplasm, in half. This is called cytokinesis, and it happens a little differently depending on the cell. In animal cells like ours, the membrane just pinches in the middle, kind of like tightening the drawstring on a bag. But plant cells have that tough outer wall, so they can't do that. Instead, they build a new wall right down the middle, called a cell plate, that grows outwards until the cell is split into 2. Pretty clever, right?
Okay, so if mitosis is the story of making identical copies to build a body, there's a very special sequel to that story, It's called meiosis, and its purpose is totally different. It's not about making clones, it's about creating variety for the next generation.
Meiosis is what we call a reduction division. It takes a normal cell with 2 sets of chromosomes and, through a specialized process, turns it into 4 unique cells that each have only 1 set of chromosomes. The whole point of meiosis is to create the sperm and egg cells needed for sexual reproduction.
The real magic of meiosis, the part that makes you genetically unique, happens right at the beginning, in a stage called prophase 1. Here, the chromosomes find their matching partner. Imagine you have 2 sets of encyclopedias, one from each of your parents. In meiosis, the A volume from one set lines up right next to the A volume from the other. And then, something absolutely amazing happens. They swap information. A chunk of one chromosome breaks off and attaches to its partner, and vice versa. This is called crossing over, and it is the key to genetic variation. It's how nature shuffles the genetic deck, making sure every single child is a completely unique mix of their parents.
So meiosis is really a 2-part deal. The first big division, meiosis I, is all about separating those matching pairs, so one parent's A volume goes one way and the other parent's goes the other. After a quick break, meiosis II happens. The second division is a lot like a regular mitosis. It just separates the 2 halves of those remaining X-shaped chromosomes. And the end result? 4 genetically unique sex cells, each with just 1 set of chromosomes all ready for fertilization.
So, we've walked through these unbelievably complex and honestly beautiful cellular stories. But what's the big takeaway? Why does all of this matter so much?
Well, mitosis is literally the reason you exist as a multicellular being. It's how you grew from that one tiny egg. It's happening inside you right now, making new skin cells, new blood cells, and lining your gut. It's the fundamental process for life's day-to-day growth and maintenance.
And meiosis? Oh, its importance is on an entirely different scale. On one hand, it's what keeps the number of chromosomes stable from one generation to the next. But its real masterpiece is creating variety. That genetic shuffling, that crossing over we talked about, creates almost endless new combinations of genes. It's the engine of evolution. It provides the raw material that allows species to adapt, to change, and to survive.
So, that kind of leaves us with a final thought to chew on. Mitosis is the process that builds and maintains your body, day in and day out. Meiosis is the process that looks past you, creating the genetic diversity for the future of your entire species. One builds the present, the other builds what's possible. So, which one do you think is more important?